Hello, my name is Jacqueline Serrati. I have my name and the date, which is 7/25/26. That's backwards. It's on there. So this is my midterm exam. The first question was, what score did you earn on the lab safety activity? I got a 95, and my favorite part was working with the fume hood, interacting with the chemicals. So that was really cool. I was never really able to work with a fume hood in high school or undergrad, so it was kind of cool doing it virtually. And I really liked how it was realistic. I feel like in class you're never really able to touch the eye wash station, and in this one you were able to see how it really worked. And I think it was kind of cool seeing like what it looked like coming at you. And I also really liked being able to read the different like safety concerns, because that's something I've always loved on the safety data sheets. So that was awesome. What score did you earn on the chemistry safety lab activity? I got a 94. I think that it says like, what could I have answered differently to earn a higher score? I think that I could have disposed of the chemicals better, and those questions that interacted with that. I kind of struggled on it virtually, so I think that that was like where I went wrong. And I also struggled putting out the fire because it was kind of hard to see everything, and if you clicked on something wrong, it kind of went dark. So being able to do it virtually was a little bit of an issue, but other than that, I think that I would have had like a higher score than the 94 if I was able to answer those questions correctly. In the scientific method lab activity, we designed an experiment and tested a hypothesis. So the experiment was about the XP cells, XP drug affecting the cells of the kidney. So I found it really cool because we were able to look at like the human body, which I love that part of science for it. So being able to basically conduct the hypothesis saying like XP is hurting the or is killing off the kidney cells, that was awesome. And we're able to use the scientific method of like finding a problem and questioning it, and then creating your hypothesis, making sure that it's testable, and then going through with your different variables, and then being able to support or reject the hypothesis. It was in supporting of the hypothesis that XP was hurting the kidney cells, so I love that as well. Describe the difference, this is number 4. Describe the difference between pipetting and micropipetting, and then describe the correct use of the 2 stops on a micropipette plunger. So pipetting is the process of moving one or large amounts of liquid from the one thing to another, so one beaker to another, using that's the standard pipette, which is typically with milliliters. And then micropipetting is transferring the small amounts, typically using microliters. So there's different stops. So the 2 stops on the micropipette plunger would be the first stop, which is when the plunger basically it's put on. So it's basically the first stop is before the tip is put into the liquid, then slowly release the plunger to draw the correct volume of liquid into the tip. And then the second stop is basically being able to blow out any extra remaining fluid from the tip. So making sure that everything that we measured is in there. The 5th question was, in the context of the human impact on climate change, we had to describe what anthropogenic climate change is, list the human key activities, and then describe the effects on the global change, the environment, and people and their livelihoods. So anthropogenic, I think I'm saying it wrong, but anthropogenic climate change is basically what is caused by humans, so the climate change caused by humans. It's the actions that we do that increase the greenhouse gases. So it can be used when we burn fossil fuels for like electricity or gas, deforestation, livestock farming, industrial manufacturing. So we see a lot of this during like the Industrial Revolution. I kind of kicked it off. And then waste removal. So like like wastewater plants, where we see a lot of like methane given off and stuff like that. A global climate change that would be affecting us would be like the environment would be like the rising temperatures, melting glaciers, more frequent heat waves, droughts, stronger storms. That if something that affects humans, like what exactly was it? People, which would be poor air quality, damaging homes and their infrastructure, and then it affects the livelihood through having water shortages, declining fishing or fisheries, increased risk of wildfires, which we're seeing a lot of lately everywhere, I feel like, and reduced crop yields. So less food available, which does affect livelihood in general for farmers and people. Number 6 was describe the difference between the chemical and physical change. So physical change just changes the appearance of what something looks like without changing the chemical makeup. So an example of this would be like ice turning to water, then water turning to a gas, like what do you call it? Like water vapor. Then the chemical change is when a substance changes its chemical makeup, basically creates a different chemical property. There's signs of chemical change through like change of color, change of temperature, change of odor, production of gas, and yeah. The simple distillation, so it also asks to describe, explain simple distillation and comment on whether the method of is whether it's a physical or chemical change. So simple distillation, it's a method used to separate a mixture based on the differences in boiling points. So how that works would be like the mixture is heated until the substance with the lower boiling point basically evaporates. Then that vapor travels into a condenser where it's converted back into liquid. And it's a physical change because the chemical makeup doesn't change itself. It just is brought from like water vapor back to liquid. Describe the structure and organization of the periodic table and focus on the main trends among groups and periods with regard to atomic properties. So the periodic table increases by atomic number, so going across, well, it would be the other way, whatever. It goes across and it's basically changes by the number of protons. The elements in the same group typically have the same number of valence electrons. So going across the period from like left to right, we see that the atomic radius decreases because when there's the more electrons, it pulls it closer. The ionization energy and electronegativity also increase as it goes across, and the elements become less metallic. So we even see this as we have like alkaline metals, transition metals, and then we go into more metalloids and then nonmetals. Going down a group, so going from basically top to bottom, we see that the atomic radius increases. We see that there's ionization energy and the electronegativity decreases. And then the elements also become more metallic. And we even see that with those lanthanoid metals at the bottom. So they become more metallic, and there's also less like non-metals that stick in that like upper right hand quadrant part of the periodic table. So we don't see that anywhere as it gets down to the bottom, even those last few rows. So, and all these components obviously dictate its reactivity in chemical compound or chemical reactions and its bond formation. The 8th question is, what is the difference between a chemical solution and what is electrolytic, versus one that is non-electrolytic? In the context of the conductivity testing lab, describe application. So basically an electrolytic solution contains ions that carry an electric current, so it conducts electricity. In a non-electrolytic solution does not produce ions in water, therefore it does not conduct electricity. So in our lab, we see that conductivity testing can be used to identify whether a solution is an electrolyte. So we test the purity of water, monitor the water quality, check the concentration of any dissolved salts, and then analyze the solutions used in like environmental, medicinal, and industrial applications. Our 9th question is to summarize the economic dynamics lab activity in your summary. So basically we were able to see how nutrients move through ecosystems and how changes in nutrient levels affect the environment. We focused on the interactions between organisms and nutrient cycling and different balances in our ecosystem in general. It shows how phosphorus is an essential nutrient for plant growth, which we've also seen in our discussion posts. It also explains that fertilizer adds large amounts of phosphorus to soil. So when excess fertilizer is washed into rivers and lakes, it can cause eutrophication, which basically increases algae growth. So describe, so as the algae die and decompose, the oxygen levels in the water decreases, which create those like dead zones where fish and other organisms can't really survive. So it demonstrates how human use of fertilizers can really disrupt the natural phosphorus cycle and harm aquatic ecosystems. Describe the carbon cycle and focus on carbohydrates and CO2 in your description. So the carbon cycle is basically the movement of carbon between our atmosphere and living organisms and even like Earth in general as a whole. So plants take in the carbon dioxide, which we know from through the air and photosynthesis, and it converts and sunlight to convert it into carbohydrates. So which that would be glucose, and the glucose stores energy. Animals and humans obtain these carbs by eating plants or other animals, and then through cellular respiration. So when we breathe, carbs are broken down and release energy. So CO2 is returned to the atmosphere, and then it's also released when organisms decompose or when fossil fuels are burned, which continues the carbon cycle. So yeah.